Let's turn now to the second part of this introduction to QSAR. In part two, we want to focus on trend analysis and filling data gaps for the endpoints that are important to hazard assessment. But first, I'd like to just recap some of the lessons that we learned from part one. We saw that the vapor pressure correlates with rodent LC50s. And we speculated that hundreds of other molecular descriptors may not. We didn't try a lot of these in this example. You're welcome to do so in the toolbox. But the bottom line is vapor pressure correlates with toxicity because it probably is involved or important indirect measure of potency in this toxicity mechanism. The endpoint. The trend in the data will be between vapor pressure and LC50 will be evident only when the toxicity mechanisms of all of the chemical is the same. Chemicals with other mechanisms, as I pointed out, acrolein, phosgenes, some of those that are very reactive chemicals, will appear as statistical outliers, generally much more toxic, to the major trend that we saw because they do not produce lethality as do the aliphatic ethers. The fact that they're a statistical outlier or just much more toxic than the rest of the group means they do not belong in the same group as the aliphatic ethers when you are evaluating lethality. The QSAR approach uses trend analysis to evaluate mechanisms in this very broad way and to test for chemical similarity in terms of common toxicity mechanisms or mode of action. In other words, the trend analysis itself very quickly tells you whether or not you have mixed modes of action or whether or not the molecular descriptor that you're using to plot the trend has any relevance with that toxicity mode of action. Simple trend analysis, looking at the data, is a crucial part to judging whether or not you are going to be able to extrapolate from one chemical to another in terms of biological activity. This principle will be used over and over and over again with many different endpoints and many different molecular descriptors that we use in a profiler in the QSAR toolbox. So we want to make sure the simple concept of trend analysis and how it can be interpreted, we want to make sure that this is very clear before you go into the use of the toolbox. And there's nothing mysterious about QSAR research. I mean, the process of developing models or mechanisms of similarity measures. It simply asks questions about why a chemical behaves the way it does through all kinds of tests. You can look at almost any measurement that can be made for a chemical and look at the chemical structure and say, why did that occur the way it did? Why are we observing this pattern or this set of measurements? And just as a simple example, so we're not thinking about a lot complex biological activity and metabolism and everything else, let's just take a simple example of boiling point in chemicals. If we if we think we know something about chemical structure and how to quantify it, then we ought to be able to understand and predict the boiling point. And even if we don't have an explicit model, we can gain lots of insights into chemical structure by just seeing if we can find some structural feature that creates the same order as you see in a list of chemicals. Let me just show this by example. In the following slide, I picked out a series of chemicals with a wide range of boiling points. In this slide, you can see that at the top is amylamine with a boiling point of about 103 degrees. And we have ethyl bromide in the middle with a boiling point of 38 degrees, and nonane at the bottom with 151 degrees. What is it about structure that makes that measurement of activity occur where it does? We may not be able to predict it absolutely, but at an initial level in QSAR and the pursuit of quantifying structure, we would just want to say, can we calculate something for each of these chemicals over on the side from their structure that would be at least ordered in the same way that you would see in this boiling point. So the first step would be, well, let's just put all of the chemicals and the variable that we're using to measure activity into ascending order, as you can see in the next slide. The goal of QSAR then would be to just calculate molecular descriptors for each of the chemicals and look for those that give the same order. If a molecular descriptor is used in this canonical ordering, it produces an order different from that of the measured melting point, then that descriptor is clearly not reflecting the right intra and intermolecular forces that control boiling point. So it's simple. If you have a descriptor and the order switches different from the boiling point, clearly there's something you don't understand about structure. So again, once you put it in order, then you began looking at properties. What are the properties that are associated with the real small chemical, or the real low boiling points, and which are the kinds of properties that are likely to be encountered for chemicals with very high boiling points. And again, just inspection of a list like this would suggest that the size of the molecule, whether or not it has hydrogen bonding groups, whether or not it has dipole moments, and so on, all of these factors vary in this list of chemicals, and a descriptor or a set of descriptors might be used to simply account for the spread in the boiling point as you change the structure of the chemical. In the next example, we might assume that we've compiled a set of toxicity values, in this case an LC50 in fish, 96-hour LC50, the common ecotox endpoint. And we've done this for chemicals which we expect to be nonpolar narcotics. If we were to pick all of these chemicals that we think are nonpolar narcotics first, and then look for toxicity values of those chemicals, we've confined them to a given mechanism. And I just want to point out that even though some of these mechanisms seem to be relatively non-toxic, the actual toxicity values or measures of chemical potency for a single mechanism can range as much as 6 orders of magnitude. In this slide, again, I suggested along the bottom that there are many mechanisms of toxicity that would cause lethality, in this case in fish, lethality in a fairly short period of time. There may be 100 different mechanisms, but we picked out just one here, called by arbitrarily chemical class or mechanism 2. This within that mechanism, if you looked at literature values, you could find a very wide range of potency values once you pick them out by class. Now, just as we have shown in the example with the boiling point, what we would try to do next is to find a molecular descriptor that created the same order in these potency values for acute toxicity. So we would embark on a series of molecular descriptors and look for this same order. In this example, we chose a octanol water partition coefficient because there is much data to show that this is the counterpart in aquatic systems to vapor pressure in inhalation, a mammalian inhalation system. But KOW, as we show on the next slide, explains most of the variance, keeps the order the same, and gives us a good trend between the LC50 and the log KOW. You can jump ahead to the next slide if you want to take a look at that, but the point is, with a good trend between the structural attribute or molecular descriptor and the biological activity for a well-grouped toxicity mechanism, the similarity of all other chemicals can be judged from consistency with this trend. In other words, plot another data point on here with its toxicity and its log KOW, and if it falls off, does not fall within the trend, it clearly is behaving with a different mechanism and should not be used in making an estimate about an untested chemical. As we'll see all through this presentation, this type of trend analysis is the centerpiece for defining and defending chemical categories later on in part 3. So just to recap on this slide of acute toxicity versus KOW in fish, this can be a regression model. It can be considered a simple trend. If we once have a homologous series of chemicals that we know have a trend, we can just by inspection look at where other chemicals fall in this line to understand whether they belong to this chemical mechanism or not. Clearly, any class of chemicals that falls off this line should not be included to estimate the toxicity for these chemicals. Pretty straightforward, but often overlooked point on using trend analysis. Now I want to show you just an example of how powerful these models can be. These are the same data points, and in this case now we've compiled about 200 chemicals, industrial chemicals. They're LC50s for fish, and we've plotted that even with some of their 30-day MATC, the no effect or maximum allowable toxicant concentration used in ecotoxicology. This is basically a chronic endpoint based on growth. And what you see is that for the same chemicals, they are parallel lines, parallel trends at a little bit lower concentration, about a factor of 12 or something like that, for this particular group of chemicals, the nonpolar narcotics. If you know that a chemical is a nonpolar narcotic, which probably includes over 50% of the industrial chemicals, which are not designed to be biologically active or toxic by any specific mechanisms like a drug or a pesticide. They're just, if they happen to be toxic, it's by accident. They're usually designed for something else and some other function, and they just happen to have some properties that interact adversely. But in general, a large number of industrial chemicals fall into this class, and if you know that it is a nonpolar narcotic, then you should be able to estimate not only the acute toxicity but the chronic toxicity for screening purposes, certainly just by using a simple log P or log KOW, the octanol water partition coefficient. Of course, the real complexity is making the decision of whether a chemical is going to behave as a nonpolar narcotic. And I wanted to show here an example of a different mechanism, in this case oxidative phosphorylase uncouplers. These are the strong or acidic lipophilic and strong acid compounds, or lipophilic anions, dinitrophenol and highly chlorinated phenols and so on. And what you can see is their LC50s are at much lower concentrations than the polar narcotics on the first slide. We get the same kind of relationship between the acute and chronic endpoints, but they're shifted down to higher toxicity at a given octanol water partition coefficient. And finally, we can find the data for a variety of reactive chemicals or electrophiles, both soft and hard electrophiles that we won't get into right now. But reactive chemicals bind much more tightly to macromolecules, and generally these chemicals are much more toxic than polar narcotics or many of the other mechanisms. These are among the more toxic chemicals. They also have many other adverse biological effects, such as mutagenicity and carcinogenicity and so on. But they can be detected very quickly on a matrix or a trend analysis of log P and toxicity. So we've covered a wide range of very broad principles, definitions and principles in QSAR, but I wanted to focus a little bit on what I see as the current limitations of QSAR, because they become important on how we apply QSAR in the toolbox. The general QSAR approach is most reliable for chemicals where the parent structure is the actual toxicant. That is, for many years, the major criticism of QSAR is that why would you use a molecular descriptor calculated for a chemical structure when it enters the body is metabolized to another chemical that is actually the toxicant, then you're using the wrong structure in the toxicity estimate. And how could you believe a structure activity model that did that? And in fact, I had a talk I gave at at a major national conference. That was the criticism from the audience. How can you expect QSAR ever to overcome this limitation of having to work with the parent structure? Well, the way that limitation is overcome, it is a limitation, but the way it is overcome is by addressing metabolism from a structure activity perspective. Predicting the effects of chemicals that are metabolically converted to more active metabolites has been a major limitation in QSAR models. Predicting metabolic activation in many test species is being overcome with metabolic simulators. These are essentially virtual livers, kidneys, lungs, plants, lots of different environments. They begin by basically creating a library of all known metabolic reactions that occur in life, and then trying to adjust the relative rates or transformation probabilities for all those reactions so that they mimic metabolism in actual metabolism experiments. Through this iterative process of trying to improve the simulation of metabolism, and once we can calculate or predict what the metabolites are likely to be, then we can take a library of toxicity models and run it on the parent compound and all of the metabolites to identify the most toxic form, and use that in predicting the toxicity in the model. A simple example will be just metabolic activation of alcohols to aldehydes. Shifts the mechanism from what might be a narcotic to one of a shift base former, but be that as it may, that is a predictable reaction, and the use of metabolic simulators just change the idea that we're dealing with one chemical to the idea that we're dealing with a family of parents and daughter products. But once you know that and you have a computerized library of models, then the actual running it on one chemical or 100 metabolites is relatively true. On this next slide, I'd just like to show an example of metabolic simulation. The top structure up here is the acetylaminofluorine compound, and when that compound is put through a metabolic liver metabolizing system, it finds three primary pathways. The simulator does. And the red color in here, the rosy color in this slide, it would indicate that these are activated metabolites, meaning they have shifted from being a non-reactive chemical to an electrophilic species. These primary metabolites are then degraded further and down, and through the various pathways there are other metabolically activated compounds that show up that could then cause effects especially in longer term toxicity. I wanted to show, I want to mention one other thing, and that is the sort of the light green colored circles there are metabolites that have been actually found in metabolism studies and have been reported in the literature. So the green part is superimposed. This is the actual measured data or actual metabolites reported from this compound, and indicate that the three pathways, first of all, that the three pathways are probably reasonable based on the experimental evidence, and more importantly that there are no other pathways been observed that the simulator has not predicted. So this is a growing field of important field in QSAR modeling, and it's addressing principally the first limitation that I mentioned about the fact that many times the toxicant is not the parent compound but a metabolite down one of the metabolic chains. A second major limitation in the state of the science for QSAR, I think, is that QSAR specialists tend to predict what they can predict rather than try to solve a hazard assessment problem. That is, if you're wondering if the chemical is a carcinogen and a QSAR specialist gives you the LC50 or the bioaccumulation factor, it doesn't really help anything. And the reason for that is that the vast majority of QSARs that are reported in the literature are reported for relatively short-term phenomena. And in fact, the general QSAR approach works for short-term bioassays, and especially well for those where steady-state exposure can be achieved. However, there are very few reliable QSAR methods or models for long-term toxic effects, and particularly the low incident probabilistic effects such as cancer. And these result from not only the chemical perturbation of some biological function or activity, biological function in the organism, but they're also conditioned or influenced by many other biological factors as well. QSAR models for long-term effects will be limited until modeling both the chemistry of the primary perturbations and the disease progression over time can be integrated. Just to recapture this again, I think generally QSAR works well on short-term assays or assays where steady-state exposure, in vitro assay, let's say, with steady-state exposure, binding assays for receptors and so on. But when you start predicting a liver toxicity in a 30-day repeat dose assay in a rat, that there are very few QSAR models in existence that pretend to do that. So this business of short-term effects and long-term disease progression in QSAR modeling is a major limitation. The bottom line is it would appear that QSAR modeling cannot really address the most important biological endpoints that are used in hazard assessment. And approach to overcoming this limitation that we are proposing is the use of an adverse outcome pathway. On this slide, I want to just give a conceptual overview or a framework for the whole idea of an adverse outcome pathway. And this has been published in the literature a few years ago as a conceptual framework for how are we going to address the prediction of long-term toxic effects. And in doing so, we want to try to separate the primary or the major classes of phenomena and processes that are happening. So in this conceptual framework, we envision a parent chemical which is put in water or introduced into the bloodstream of an animal or the gut that goes through a series of chemical speciation steps, metabolism, interacts with a whole series of reactions. And that process is something that chemists and biochemists should be able to model. In other words, we're converting the parent chemical to a number of different chemicals, either from metabolic activation or just protonation, tautomization, all of the speciation changes when you dissolve a chemical in water or another biological fluid. That is modelable, and some of those interactions with the solvent and biological molecules are considered molecular initiating events. In other words, when we bind to a particular protein or to a DNA or to a receptor site or something else, some of those cause a perturbation that initiates a biological effect. And those are called molecular initiating events. If we look at all of the receptors that are located in the body, the different and all of the non-specific binding sites on proteins and DNA, and it's finite, then we should be able to adequately account for the chemical interactions using QSAR to predict this chemical, a particular chemical, will lead to a family of molecular initiating events, one or more. Most often, a chemical will bind to many, many, many sites. The consequences of that depends on how you observe biological, what species, what time delay, how you do the exposure, and so on to get the array of biological activity. But the main point here is that leading up to the molecular initiating event, that part is where structure activity does well. Now, when you have a molecular initiating event at the molecular level, there are a series of biological responses that are unique perhaps to some species, but in many cases the responses to let's say inhibition, binding to the ER site in an organism or a cell, those things are highly conserved across many biological species. But you have a series of biological responses that would be at the subcellular level, cellular level, organ, tissue, into the whole animal. And there are bioassays that can measure those effects, and some of those are considered to be adverse outcomes or are useful endpoints for hazard assessment, hazard identification, hazard assessment, risk assessment, whatever we want to do. So we have a cascade of effects here separated in the middle by a molecular initiating event, and we are approaching this problem or this limitation by saying the job for QSAR is to really work on the left side of the slide, conceptual framework, and then we have to work on how are we going to do the systems models or the biological response models that interpret and those basic events in molecular perturbations and cause adverse biological effects. Let me just go on and describe this in a little more detail. In this next slide, again, we are looking at the individual toxicant and trying to come up with the chemical reactivity profiles. The reason that we've used this terminology is that you will see that there is a profiler, a chemical profiler in the toolbox. And the intent of the toolbox is not to become a mysterious thing, but really to automate so much of the detailed chemistry and biochemistry so that not every hazard assessment person has to know that. What he has to realize is that when you put in a chemical toxicant, there are a large library of models that can be run to create a profile on what kinds of molecular initiating events this chemical can cause. So these reactivity profiles link up to a whole series of receptors, different nucleophilic sites on DNA, the softer nucleophilic sites on proteins such as amino groups and thiol groups. And we look at the chemistry and biochemistry of all these reactions to basically make a mechanistic profile. What you will see in using the toolbox is that many times judging the similarity of the chemicals in a class to decide whether or not you can extrapolate from chemicals to chemical, group chemicals, do trend analysis. Many of the considerations for grouping chemicals and judging similarity can be done with this mechanistic profile here on the left side of the screen and nothing more. You can evaluate how accurate your judgment is using the biological data, because the biological data will not lie. If they don't follow a good trend, then there are more than one mechanism happening, and the answer to that is probably back into how the chemical has been profiled. But the mechanistic profiling is done here on the right side, leading up to a group of possible molecular initiating events given for a specific chemical. On the next slide, we are showing that from these molecular initiating events, we might look up away from the molecular interactions to cellular observations of biological effect of those perturbations. In other words, we would then look at gene activation assays, protein production, vitellogenin production for estrogen signaling pathways and so on, or signal alteration at the cellular level. There are a whole series of assays and that can be used to decide which ones of these molecular initiating events are actually most important for this chemical, and can that be predicted? Just some of you have seen the National Academy of Sciences toxicity testing for the 21st century document. When they talk about a toxicity or toxicological pathways, they're generally talking about the molecular level interactions and how this the molecular biology of this. The adverse outcome pathway is really saying, how can we take these molecular interactions that occur at the cellular or subcellular level, and how can we then measure how the ramifications of those perturbations as they progress at different levels of biological organization. In the next slide, we might extend this same process out to at the organ level to look at specific organ function or developmental effects on various organs in different life stages. And again, what we have seen is that a large amount of the high throughput screening and the in vitro assays tend to be focused in this particular region of going from molecular initiating events to organ effects. And finally, in the last slide in this series, we look all the way out to whole organism levels of lethality, sensitization, whether it be skin or lung, birth defects, reproductive impairment, cancer. At the organism level, or if you look at ecological risk assessment, of course, the population level effects are often more important from a regulatory point of view, where you look at just the structure of the population or extinction of individual organisms. But this is a conceptual overview of the adverse outcome pathways. The reason we're concerned about this again is because predicting how biological responses cascade through different levels of organization is not a structure activity problem. It's a systems biology problem. It requires a group of biological response models, or at least calibrations of taking a short-term lethality measure and calibrating this all the way back to a molecular initiating event. Sometimes you will have a threshold. If you cause this much damage at this level, and more than that, the animal will die. That's a very simple threshold that basically calibrates all the way through these different levels of organization. But there are groups working on how do we link these two areas of research together: the QSAR mechanistic profiling and the biological response models of systems biology. That work will continue on and probably will overcome the limitation that we've been talking about for the QSAR. But the take-home message here in this particular overview is to focus down here on what we described as the mechanistic profiling. And you don't have to understand how to do all that in terms of chemical theory. What we need to do is understand that there will be a profiler in the toolbox, and that profile will include the state-of-the-art chemistry and biochemistry, and it will provide you a series of possible interactions and different molecular initiating events for a chemical. And all you need to really realize from that is that if chemicals have dramatically different profiles, you should be cautious if you try to consider those chemicals as being similar to each other, even if they belong to the same class of industrial chemicals. I think that covers the main point for the adverse outcomes. But just to take this one step further and illustrate in advance of your training on the QSAR, on the Toolbox, let me just spend a little bit more time on some of the interaction mechanisms, and so that the terminology that you hear early in the course in the Toolbox training is not a mystery. If we were to just focus now on reactive chemicals, and we would pick in these reactive chemicals those that covalently bind to nucleophilic centers. These interaction mechanisms form covalent bonds, so they permanently bond to thiol centers, amino centers, all kinds of nitrogen, oxygen centers in DNA. There are a large number of interaction mechanisms. For example, Michael addition is a type of interaction that groups chemicals with double bonds, and especially activated double bonds, can undergo. There is shift base formation, which is basically a reaction of a carbonyl with an amino group on proteins or even on other macromolecules. And there are names for these different kinds of reactions. Without getting into the details of the chemistry, you just need to understand that these are different kinds of interactions, but they all produce one kind of interaction, and that is what you'd call an atom-centered irreversible covalent binding. In other words, they're going to form a permanent new molecule and cause a permanent perturbation, not a reversible, necessarily a reversible interaction. So even though you might consider the chemicals to be an electrophile, that doesn't control the biological effects. What controls the biological effects are the different sites that it binds to. Is it at a nitrogen group? Is it at a sulfur group? Is it at the DNA? And excuse me, and a number of different mechanisms will have different rates, and therefore it would show perhaps as a potency value change. By understanding these interaction mechanisms, you should be able to predict whether you would produce protein thiol adducts, glutathione oxidation, glutathione depletion, amine adducts on lysine derivatives or other free amino groups, perhaps a ring nitrogen or other substituted higher order nitrogen adducts, DNA adducts. There would be a profile. The interaction mechanism profile would lead to a profile of adducts and basically damage form in the biological system that we're putting it into. The problem of predicting the consequence of forming these array of adducts is not a structure activity problem. It's a biological response problem. So that if you basically deplete glutathione by having a high enough concentration of the chemical there to do it quickly, you produce oxidative stress in the cell in almost all organs that the chemical gets into. If in many of the cases these same chemicals that can bind, let's say, to a protein and cause protein sulfuratics, these will appear as irritants, but they can also appear or cause skin sensitization by, we've noticed in one of our series of workshops that basically identifying skin sensitizers are those that can produce antigenic compounds, which is really producing a protein addict. And we began separating out the different kinds of biological effects that different interaction profiles can give us. I think this is enough of an overview, and I'm just hoping that this explains some of the terminology that's used, and more importantly, why we find this so important in just grouping chemicals so that we can look at trend analysis and read across methodologies to fill data gaps.